Okay, so if you already have done this part, you can fast forward, okay, because we did this part in class. So, but if you were absent and you don't know what to do, we're going to start off by reading this part right here. Okay, so the genus Paramecium, recognize that Paramecium are unicellular organisms, and they're like a notch above bacteria, to be really honest with you. They are often found in ponds, and so if you get like a teaspoon of pond water and put it on a microscope, you'll see a lot of these guys. Under ideal, like under the best conditions, which means sufficient food and water and space, their populations grow rapidly. They grow rapidly, okay? And so we call that exponential growth, okay? That means a very high biotic potential. And so that graph looks like this. Oh, sorry, that's ugly. Okay, so it's what we call a J-curve. Okay, so their population just goes up and 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 up. Okay, under the best conditions with the most food and water and space. These conditions right here, the food, water, and space, those are density dependent factors. Density dependent factors. Okay. The only time it becomes density independent is if we're talking about the weather or we're talking about geological events like earthquakes or anything like that. But food, water, space, mates, nesting sites, nutrients, sunlight, all of that stuff, essentially organisms are going to fight over that. So anything that organisms are going to fight over or compete over is going to be density dependent. Alright, so here's your definition for exponential growth. So circle your keyword, and that is a growth in which the total number of potentially reproducing organisms increases with each generation. However, populations of organisms will not increase in size forever. So what do I mean by that? Eventually they're going to run out of food or water or space or mates or whatever. So because their population is getting so big. So they're going to start off in that J curve. And then they're going to go down and up and down and up again. Okay, over and over and over again. And that stabilized population right there is called the carrying capacity. Okay? So when the population size is stable, that means that the population has reached carrying capacity. Alright, and so here is the definition of carrying capacity. That is the number of individuals that an environment can sustain without being degraded. So competition for resources among members of the population. So if it's among the members of the population, so if we have like a pride of lions and they're fighting over food, they're all the same species, that's called intraspecific. That's called intraspecific competition. So it's within the species. Okay? If it's between different species, like if we're talking about the lions and we're talking about the hyenas, so lions and hyenas, something like that, between different species and they're fighting over like the zebra or whatever, that's going to be called inter-specific competition. Okay? So that's going to obviously affect population size. So anytime we're competing, we're going to be affecting population size. In the classic series of experiments in the 1930s, Russian ecologist, so Gauss, this guy right here, he formulated the principle of competitive exclusion. So there is another term right there that you should know. What that means is that if you put two species in the same place at the same time, with the same food and the same niche and everything, then they will begin to compete with one another. And the one that's more fit is going to survive, and the one that's less fit is going to decline. So their population is going to decline. So if two species are competing for the same resource, the species with the more rapid growth rate will outcompete the other. In other words, no two species can occupy the same niche. Okay? So not just rapid growth rate, but also most fit. So the species that's most fit for the environment will survive and reproduce. Okay? So that comes from Darwin. Remember Darwin and natural selection. In competing populations of organisms, genetic variations that reduce competition, so any genetic variations, any adaptations that reduce competition are favored through natural selection. And again, there's another key word right there, natural selection. That's your Darwin. Okay? Suppose two species A and B compete for the same food source. Individuals of species A can also use another food source. This is going to reduce the competition over the food source needed by species B. The individuals of species A can use another food source and survive because they do not have to compete with species B. Okay, so the less competition, the more survivability. So less competition for resources, the more an organism or population can survive. In nature, organisms frequently invade unoccupied habitats simply to avoid intense competition. So organisms frequently invade in order to avoid intense competition. Once an organism is in a new habitat, any variations that allow it to use its available resources will tend to be perpetuated through the population. What do I mean by that? Those variations, another word for that is adaptations. So anything that that organism or that population has that allows it to use the available resources is going to increase. Perpetuate means to increase through the population. So you're going to see more of those. Darwin said that. So in this way, eventually the genetic makeup of the population will slowly change and become adapted to its new niche. Consider the example of Darwin's finches. Okay, so remember the finches all had different beaks because they all ate different stuff. Alright, so what you're going to do next is you're going to write a hypothesis or a prediction. It's also a prediction, so you're going to predict. So you're going to write two sentences. So the first sentence, what's going to happen if you put the two paramecia in two different test tubes in their own test tubes? So what happens if they grow alone? Okay, what's going to happen to their populations if grown alone? And then the second sentence, you're going to predict how are they going to grow when they're grown together. So what's going to happen? Okay? So two sentences. I should see two sentences. Alright? So we're going to go up to the top. If you're asking me what we did today, this is it. So you can sit down and do it. I'm recording. Thank you. Sorry. Alright, so the purpose, so after you write your hypothesis, your two sentences right here, the purpose of this lab is to test the principle of competitive exclusion. Okay? And if that exclusion, if this principle is accurate, our purpose is to determine the carrying capacity of the most fit paramecium. Okay, that is the purpose. So, what you're going to do is you're going to count these cells. So here's your P. aurelia all by itself in its own test tube. There's only one. Okay? So right here, P. aurelia grown alone. There's only one, but it wants you to multiply by 2 every single time. So 1 times 2 is 2. And then P. caudatum all by itself in its own little test tube right here. Again, there's only one. Okay, so P. caudatum grown alone. Okay, and it wants you to multiply by 2, so 1 times 2 is 2. And then in the mixed culture, so this is the third test tube right here. You're going to count again. This is 1 P. aurelia, 1 P. caudatum. So there's 1 P. aurelia, 1 times 2 is 2, and then 1 P. caudatum, 1 times 2 is 2. Alright? And then you're going to go on to day 2. Okay, so here's day 2. Here's P. aurelia all by itself. And so there's 1, 2, 3, 4, and 5. So 5 times 2, because you have to multiply everything times 2, so that's going to be 10. And then 1, 2, 3, 4 of the P. caudatum right here. So 4 times 2 is 8. And then in the mixed culture, we've got a little bit more going on here. So these right here, the P. aurelia, so I've got 1, 2, 3, 4. Got to multiply it by 2, so that's going to be 8. You make sure, yeah. Alright? And then P. caudatum, I've got 1, 2, 3, 4, and 5. Multiplied by 2, there's 10. Okay, and then I'm going to go on to day 4. I think you're getting the hang of it. So day 4 should look, if I count it correctly, hopefully I did, it should look something like this. 52 of the P. aurelia by itself, 26 of the caudatum by itself, 34 of the aurelia in the mix, and 22 of the caudatum in the mix. And hopefully I counted that correctly. Now you're going to finish the rest of it, so you're going to do the rest of your counting. And then when you're done with the rest of your counting, okay, then you're going to graph. Alright, so this is where everybody should be caught up. Alright, so here's your graph, here's your x-axis, right here going across, here's your y. Okay, so your x-axis is going to be the time. And that's going to be in days. Okay, so remember they went by 2s. We have 0, and then 2, 4, 6, 8, 10, 12, 14, and 16. Okay, so that's time in days. And then here's the number of cells. That's what you're going to name the y-axis, number of cells. Okay, that's what you counted. And so typically remember that your dependent variable, whatever it is that you're counting or measuring, it's going to be your dependent. Okay, I don't know that anyone has cells that are higher, a number that's higher than 100. So I think it's pretty safe to go up by tens here. A little higher than I should have, but there we go. Alright, so that is what your graph should start off. And so you're only going to graph the last two columns, these two right here, the last two columns. So you're only graphing these two columns. This is your X, these are your Y's. So you're going to end up with two lines on the same graph, which means that you've got to have two different colors, right? You've got to have two different colors because you have two different lines. Alright, so I'm going to make my P. aurelia red. So P. aurelia is going to be red for me. And then P. caudatum is going to be green. Remember, these are just the mix. That is all you are graphing. So you should only have two lines. Alright, so on day 0, going up here, on day 0, I had 2 aurelia in my mix. So my aurelia is red. So on day 0 I had 2, and I'm just going to kind of guesstimate. I'm just going to kind of guesstimate about right there. Maybe that's 2. Okay? Okay, and then I had 8 on day 2. So on day 2, I'm going to go up to 8. So about maybe right there. Still under 10. Okay? And then on day 4, I had 34. So I had this huge jump on day 4. So I'm going to go all the way up to 34. Now I'm going to stop there. So I just kind of started it with you. You're going to finish the whole thing. Make sure you connect the dots. Okay, let me start P. caudatum for you as well. On day 2, for P. caudatum, on day 0, I had 2. Okay, which was the same as P. aurelia, which means that they're going to be on top of each other. Okay. On day 2, I had 10. It's going to be a little bit higher right here. Okay, and then on day 4, I had 22. So about right there. And then I'm going to connect those lines. Okay? Now again, I'm stopping on day 4. You go all the way across. Okay? You are required to come up with a title for this. Okay? Also, you need to label exponential growth. And so obviously you're only going to label that on the line for the paramecium that won, essentially, that lived. Okay, so you don't have to do it on the one that didn't live. Okay, and you're going to also label the carrying capacity Again, only for the most specific. I mean, not the most specific, I'm sorry. The most successful. Okay, so only for the most successful species. Okay, so focus only on either P. aurelia or P. caudata, whichever one is most successful. The other one, leave it alone. Okay? So you're going to label both of these. When you are done, you're going to answer questions 2 through 7. Okay? And you're going to answer them in complete sentences. You are not going to do this part. You are not going to do that part. Okay? Alright. When you are done, make sure that you've turned it into the tray to be graded.